Hello and welcome to another one of my videos. In this one I'm going to be looking at the polymerase chain reaction test, the PCR test for testing for viruses. I didn't know much about it before, and so I went and looked at it to see how it works and whether you can trust it. Recently, Tanzania, the president of Tanzania, did tests and faked people's names with a pawpaw, a goat, sheep, bird, and a number of things. And lo and behold, the pawpaw, the papaya, and the goat tested positive for the common cold number 19. And so it really does bring into question whether it's accurate or not and whether it can be trusted. So I thought I would go and check it out and see how it actually works to see whether it can be trusted. So even though it's a very, very clever system, and I was most impressed by the cleverness of it, it relies on a lot of suppositions and inaccuracies, which are very glaring if you actually use some logic and use your brain, to the point where you really can't trust it. So let's have a look at it and see why. Now let's look at a very brief and condensed look at what the polymerase chain reaction test actually does. Polymerase is a protein which builds the molecule, and this is the chain reaction, and it's a thermocycling reaction. It's done with heat. The DNA molecule has 4 molecules, thymine, cytosine, adenine, and guanine, and they pair. Thymine will always go with an adenine because of a bond, whether it's 2 or 3 electron bonds, and cytosine always goes with guanine, and adenine with thymine, guanine with cyanine, adenine with thymine, thymine with adenine, cytosine with guanine, and so on and so on. And like 30,000 of those long. What the polymerase chain reaction cycle does is applies heat, a thermocycling. It gets up to 90 something degrees, and that breaks those bonds between the 2 sides of the DNA molecule, and you have 2 of those strands into RNA instead of DNA. Then they cool it down and they use a primer, which string of DNA which they have manufactured, and for example the opposite of that, they will have exact copy of that in A, G, T, C, and those are only about 18 of those long, but they get them by cooling them down. That primer will attach itself to an opposing set of nucleotides, as they call them. Then the polymerase, which is the protein, goes along and reads the next one, and this is an adenine goes with thymine, a thymine with an adenine, a cytosine with a guanine, and a guanine with a cytosine, and joins them into the chain. And then in the next thermocycle, they heat it up again, which then breaks those bonds again. And look, you've got another copy of the RNA there. The same happens in the opposite, is they do the opposite version to get copies of this one as well. So it multiplies exponentially. Every time you do a heat and a cool, it will make more of these. And those copies will make more copies, and those copies will make more copies, and it's exponential, and you get these strands. It's very clever. But as clever as this is, it's full of suppositions and therefore is faulty. The first inaccuracy or supposition that I can see, which is very glaring, we'll start at the beginning of the process and then work through the process to find the errors. The first one is the very sample that you're taking. That sample is errors. Now, the media have reported that some are saying that there are inaccuracies in that. The actual taking of the samples is pretty simple. It's basically a swab. Stick a cotton bud up your nose, your throat, your butt, any hole that they can get any sample from.
there's not a lot that can go wrong with that. You can contaminate it from, say, you put it on the bench and there was a dog poop there and that contaminates it. Well, I don't think that happens very often. The big error is not in the way they take the sample, but what actually are they sampling?
Now, what they're actually trying to collect is some genetic material from your human being. Now, it's any genetic material of anything, of any kind that they can get with their cotton bud swab. Now, notice inside the human being, outside the human being. Here we have the mouth, and we have the nose, and we have the butt and everything else like that. And sometimes they do the eyes. Now, you'd think that those are actually in the human being. They're not. These three are part of your immune system. The tears that come out of your eyes are actually trying to flush out bugs and viruses, and the same with your nose. The snot that comes out of your nose is part of your immune system of trying to flush out, kill and eradicate anything before it gets into your body. Same with your mouth. The saliva that comes from your mouth is full of bacteria and things that are trying to kill bugs and things before they get into your system. And when they go down from your mouth through your intestine, there are lots of bugs and things. There's hydrochloric acid there in your stomach that kills off a bunch of bugs and things. And there are other things that kill off bugs. Your mouth and intestinal tract, your tears and your snot are all part of your immune system, which is valiantly trying to stop bugs getting into your body. When you have a sore throat, that is a failure of the saliva and the immune system to stop those bugs, and those bugs start to infect your throat and makes it sore, and it can get into your blood system and cause those symptoms. Now, the fact that you have bugs and things in your snot, your mouth, and your saliva and your eyes, which is part of your immune system trying to eradicate those, does not, even though they are present there in your nose, eyes, mouth, and intestines, it does not mean that they are resident. It is the same sort of idea as if you take your car to the car wash and you wash the windscreen, the wipers, you wash this down, and all this water runs off into the sump, and you take a swab from the windscreen, from the bonnet, the bumper, and everything, and from the sump here to find out what mud and stuff is inside your car. The fact that you have in your human being some bugs in the snot, your eyes and your mouth, and your intestinal tract does not mean that you actually have an infection in your system. It just means, like the car, that there are a lot of them around, but they are all possibly coating. It does not necessarily mean they are inside your system. The first fault of collecting this material is not the way it's collected, it's what you're collecting. You're collecting a whole heap of stuff that is outside of the body of the person, not inside. The best way to do it is to take a blood sample with a syringe or something, which is way better to take a blood sample and sample that to know what is actually in your blood. It's the most common for the PCR test is the swab, which is testing for material outside of your body, not inside.
So the first flaw is that you're actually testing stuff that isn't even in your body. So that's crazy. The next one is, okay, even if you took a blood sample and even if you trusted the material that's in the saliva because it might have leaked out of your body or your snot or something, and say you do have something inside your body that's leaking out that you're testing for. Okay, maybe. So what are you collecting and how are you going to test it? There are two main things that I find difficult with this one. First off, you're taking any kind of DNA, any genetic material from anything, from your own, from other viruses, bacteria, bacteria that you have within you, that's genetic material within the bacteria within you, the genetic material of parasites that might be there, and all of this stuff. It gets mashed up into millions of little bits and then put it into a big soup to then put into the PCR machine to then start testing for it.
Here we have some DNA. This is a nucleotide, and we've got guanine pairing with cytosines, adenine with thymine, cytosine with guanine, thymine with adenine, and they start to pair up and they go along the sequence. Then they're going to go on and on and on and on, and there's 150. There we have a line of 250 or thereabouts, and that many on the board is approximately 2,000. So you're looking at 15 times that many nucleotides in the sequence. And the only difference between the SARS-1 and the SARS-2, that's the common cold 19, is that many. That many nucleotides different in 15 times what you see on the board there.
So as you can see, there are a lot of nucleotides in a virus, 20 or 30,000. Some viruses get down to 8,000, I think, but these ones we're talking about in the 30,000 range with only about 12 nucleotides difference. And so if you are testing for anything other than the 12 of the differences, a SARS-1 can test positive as a SARS-2. When you consider that there are 4 major groups of coronaviruses, and of those 4, there are about 4 in each of those, only 6 of those actually can infect human beings. And of those 6, there are something like 37 different strains. And when you're talking strains, that's a mutation of a previous one with only 1, 2, 3, very few actual changes in the code to make it a new strain. So you're looking at viruses that are very, very similar.
Like with many things, a virus has structure to it that allows it to do the things it's going to do. It has RNA or DNA strand. It has pieces of code, obviously, and it has segments. And when they have studied those, they find that there are certain parts of the code that do certain things. One part allows it to, say, to get around. Other parts do things like convincing the cell to do something. And another part is to do with communication or whatever. And there are segments that they must have to be able to function. It's like a house. You've got to have some walls, a roof, a floor, and a window and a door, for example. And all of the houses will have these to make it a house. And there will be variations house to house. The virus also needs certain basic mechanism that are going to be exact copies of other viruses to allow it to get around and function. And there will be variations, like the difference between the SARS-1 and the SARS-2, the common cold 19, is that these 12 nucleotides allows it to be able to get into cells that it normally wouldn't be able to. And so if you test for anywhere that is within the structure of that is common to every virus, then, and even stuff that is common to other bacteria and our own system, then you will get a positive identification on something that is not.
So that's a great difficulty. There is sufficient similarity between the viruses that if you test for one area that is common to various ones, you'll get a false positive. The next flaw that I could identify is the actual primer that they make to be able to start their test. They do that by in the laboratory, getting the same thermo situation to get the adenine, thymine, guanine, cytosine to link one after another, and by adjusting the temperature they can decide which ones link after the other. Now if they have any mistake or inaccuracy in their temperatures, they're going to get false joints, they're going to get wrong pairing. And also, who decides which piece of the DNA or RNA that you're going to test? That could be either inaccurate, or they could even be blatantly lying to you, or they can select a part of the DNA strand that is common to quite a number of different viruses, so that you can get a whole bunch of viruses all testing positive to the one that you want it to be testing to. So the actual building of that primer is also suspect. Then after that, which I didn't investigate, is how you read all of that information. That's a subject that there was enough of this, and this video is getting long enough for me to basically say, well, that's enough to convince me. But I'm sure just on the face of it, the reading of the DNA, the process of reading molecules that are so small that you can't even see them, the whole entire length or size of a virus is like 8 to 40 nanometers, and you're talking individual molecules, you're getting down to atomic levels. How do you read those? And so there's bound to be some inaccuracies in that, even if you amplify it. You're also going to have problems with the amplification, because if you make a mistake in your primer or your temperature in your thermocycling in the PCR test, you're going to get wrong sequences. And if you do that early on, you're going to amplify a mistake, because there is a sensitivity of only 1 or 2 degrees of temperature, because that will make an adenine or bond with a cytosine or a guanine instead of the thymine, because the bond is a double or a triple, and if you electron bond, and if you get the temperature wrong, you can force one of the molecules to join to the other without having the appropriate bonding. So you can get a 3 joining to a 2 and a 2 joining to a 3 instead of a 3 to a 3 and a 2 to a 2. So you get your temperature wrong due to any number of factors, and you do that early on in your cycle or anywhere through the cycle, and you're going to amplify mistakes. And if you get that wrong in your primer, you're going to amplify a primed mistake. And if you amplify a piece of the virus which is common to other viruses and genetic material, you are also going to get mistakes. So left, right, and center. It's a clever method, but it is absolutely flawed. And I would never trust it, ever.